Today we are here to discuss about how the amino acid metabolism take place in our body or animal body, whatever, in plant body or any other microorganism. So as we know, amino acid are the last one, or we can say that there are 4 types of biomolecule in biochemistry. So amino acid or protein is the last biomolecule which provide the energy. The amino acid are the final class of biomolecule that make a significant contribution to the generation of metabolic energy. Amount of energy obtained from amino acid of dietary protein or from tissue protein depends on type of organism. Suppose if the organism is carnivorous, in case of carnivorous organism, it take 90% of energy from the protein metabolism, and in case of herbivorous, they need only a small amount of their energy from amino acid. Plant rarely use amino acid for energy. In plant, amino acid concentration is regulated through biosynthesis of protein or nucleic acid or other molecule needed to support growth. In animals, amino acid undergo oxidative degradation in 3 different metabolic conditions. The first condition is that during normal synthesis and degradation of cellular protein, some amino acids that are released from protein break down and are not needed for new protein synthesis undergo oxidative degradation. It means when the protein is metabolized, or we can say in case of metabolism, the protein is catabolized. In that condition, the breakdown of protein is take place into the amino acid, and if these amino acids are not taking part in the biosynthesis of other biomolecule, in that condition, the amino acid go through the oxidative degradation. The second condition is when a diet is rich in protein and the ingested amino acid are excess in body, then they catabolize because there is no process or no any storage condition for the amino acid in our body. And third condition is that during starvation or untold diabetes, whenever the carbohydrate is either available or unavailable or properly unutilized, then cellular protein is used as a fuel. Under all these conditions, amino acid lose their amino group from alpha keto acid. The alpha keto acid undergo oxidation to CO2 and H2O or enter into gluconeogenesis. If we will see the overall process, how the metabolism of the protein or catabolism of protein, a breakdown of the protein in case of the mammals, then we will see the protein, maybe intracellular protein or a dietary protein, after the digestion, it will convert into the amino acid, and this amino acid transfer its amino group from the alpha ketogluteric acid and convert into the glutamate and release the NH4, and this ammonia is used either a biosynthesis of amino acid or biosynthesis of nucleotide or biosynthesis of biological amines, and the remaining carbon skeleton is which is converted into the alpha keto acid is enter into the citric acid cycle, which is generally take place for the energy production and completely degrade into the CO2 and H2O and release the energy in the form of ATP. And this, if the ammonia is using in the biosynthesis process, then it is okay. Otherwise, it is converted into the carbamyl phosphate and enter into the urea cycle. And if we will see that this urea cycle and the citric acid cycle are interconnected between each other through the aspartate arginosuccinate shunt of citric acid cycle. What will be the metabolic fate of the amino group? If we will see this diagram, through this diagram, we can say that the amino acid from the ingested protein, which is coming through the dietary protein, is converted into the amino acid through digestion, and this amino acid will transfer the amino group from the amino acid to the alpha ketogluteric acid and convert into the glutamate, and this glutamate again convert into the alpha ketogluteric acid and release the ammonia, and this ammonia is utilized by urea cycle or utilized by the biosynthetic process. One more thing is that also glutamine also play an important role in the amino acid metabolism, or we can say protein metabolism. This glutamine is also converted into the glutamate and release the ammonia, and this ammonia is again enter into the urea cycle. And from different tissues, like if you will see in case of the muscle, the ammonia which is generally degraded through the protein. In case of the muscle cell, then it is transported from muscle to the liver, because the complete degradation of ammonia or the urea cycle take place in the liver. So the transport is necessary from the muscle to the liver, and this transportation of amino group from the muscles to the liver is taking place through the alanine cycle, which we will discuss later on. And from extrahepatic tissues, the glutamine is synthesized for the transportation of the amino group from the extrahepatic tissue to the liver, and when the amino acid is lose their amino group from the amino acid, then it is transported to the glutamate and converted into the glutamine, and this glutamine is transported to the liver, where it is completely enter into the urea cycle. Glutamate and glutamine play is especially critical role in nitrogen metabolism, acting as a kind of general collection point. If we will see the back in this point, then we can say that glutamate and glutamine are especially playing an important role in case of the urea cycle, and we can say that these are the collection point for amino groups. In the cytosol of hepatocytes, amino groups from most amino acid are transferred to alpha ketoglutarate to form glutamate, which enter mitochondria. Now finally, the transport of the amino group from all the extrahepatic tissues or muscles or any other kind of tissue transport to the liver, where it is totally converted into the glutamate, and now this whole process is taking place in the cytoplasm. After this glutamate synthesis, when the collection of the glutamate completely taking place, then it transported to the mitochondria from the cytosol, where it is then enter into the urea cycle, which enter mitochondria, it gives up its amino group to form. Now before entering into the urea cycle, this glutamate, after entering into the mitochondria, is first lose its amino group from the glutamate and again convert it into the alpha ketoglutaric acid, and this alpha ketoglutaric acid enter again into the TCA cycle. In skeletal muscle, excess amino group are generally transferred to pyruvate to form alanine, another important molecule in the transport of amino group to the liver, as we have already discussed. The one important enzyme is generally taking place when amino group is transferred from the amino acid to the alpha ketoglutarate, and this enzyme is known as aminotransferase, which is generally convert this amino acid into the alpha keto acid, and alpha ketoglutarate convert into the glutamate. Now if we will take the diet, and diet is the protein rich, then first of all digestion will take place, and the protein digestion is known as the gastrointestinal digestion, because gastric juice and the intestinal enzymes and hormones generally take place or participate in the digestion of the protein, and will convert this protein into small units, which is generally known as amino acid. And some hormones are also participate in this digestion, like gastrin, secretin, and some gastrointestinal enzyme, which are trypsin, chymotrypsin, or carboxypeptidase A and B. Pyridoxal phosphate participate in the transfer of alpha amino group to the alpha ketoglutarate. Actually, the enzyme which is generally transfer the amino group from amino acid to the alpha ketoglutarate is using a coenzyme, which coenzyme is known as PLP, pyridoxal phosphate. The first step in the catabolism of L amino acid, once they have reached the liver, is removal of the alpha amino group promoted by the enzyme called aminotransferase, because the transfer of the amino group is taking place. That's why this process we are saying it aminotransferase, or the transaminase. In this transamination reaction, the alpha amino group is transferred to the alpha carbon atom to the alpha ketoglutarate, leaving behind the corresponding keto acid. The effect of transamination reaction is to collect the amino group from many different amino acids into the form of L-glutamate. The glutamate then function as a amino group donor for biosynthetic pathway or for excretion pathway. Then glutamate will decide whether the ammonia, which is generally released from the glutamate, will take part in the biosynthetic process or will take part into the urea cycle. All aminotransferases have the same prosthetic group and the same reaction mechanism. The prosthetic group is pyridoxal phosphate, the coenzyme form of pyridoxine or vitamin B6. After entry into the mitochondria, this glutamate again release the amino group and again convert into the alpha ketoglutarate with the help of enzyme alpha glutamate dehydrogenase. This is the only key enzyme which generally utilize both NAD plus or NADP plus. This is the unique feature of this enzyme. As we have seen, amino group from many of the alpha amino acid are collected in the liver in the form of amino group of L-glutamate molecule. These amino group must next be removed from glutamate to prepare them for excretion. The hepatocyte glutamate is transported from cytosol into mitochondria, where it undergo oxidative deamination catalyzed by alpha glutamate dehydrogenase. It is the only enzyme that can use either NAD plus or NADP plus. Now finally, after when the ammonia produced by all these deamination processes, either it is take part into the biosynthesis process or enter into the urea cycle, the alpha ketoglutarate formed from glutamate deamination can be used in the citric acid cycle or for glucose synthesis by gluconeogenesis. Now glutamine transport ammonia in the bloodstream. Ammonia is, as we know, ammonia is quite toxic to animal tissues in many, including the brain. Some process such as nucleotide degradation generate free ammonia. There are so many processes through which ammonia is produced in extrahepatic tissues. In most animals, much of the free ammonia is converted to the non-toxic compound. So if there is a need of transport of ammonia from these extrahepatic tissues to the liver, then there is a need of non-toxification of this ammonia. So this ammonia first of all will convert into the non-toxic compound, and these non-toxic compound before export from the extrahepatic tissues into the blood and transport to the liver or kidneys. The free ammonia produced in tissues is combined with glutamate to yield glutamine by the action of enzyme glutamine synthetase. This require ATP. And occurs in two steps. Finally, when if we will talk about in case of brain, then brain will convert the protein into the amino acid, and these amino acid will lose their amino group in the form of ammonia. Then this ammonia is combined with the help of glutamate and converted into the glutamine with the help of enzyme glutamine synthetase, and this use energy in the form of ATP, and this ATP is converted to the ADP through the two reaction process, and finally it is converted into the glutamine. And when this glutamine is formed in the extrahepatic tissues, then this glutamine is transported to the liver in the with the help of bloodstream, and finally it reaches the liver, from where it is again lose their amino group, and then this ammonia is utilized by the urea cycle. Glutamine is a non-toxic transport form of ammonia. It is normally present in blood in much higher concentration than other amino acids. Since we are knowing that from most of the extrahepatic tissues release their ammonia in the form of glutamine, so we can say that in bloodstream there is a higher concentration of this glutamine than other amino acids. Now, if we will talk about in case of skeletal muscle, in case of skeletal muscles, the ammonia is transported from muscles to the liver in the form of alanine. As the graph is indicating, as we know that in muscles the glycogen metabolism also taking place, where the glycogen is converted into glucose, and glucose is converted into the pyruvate with the help of glycolysis. And then finally, when this ammonia is converted into the glutamate, this glutamate combined with the pyruvate, because there is a excess amount of pyruvate in muscle through the glycolysis, and by combining with pyruvate, this glutamate and pyruvate will convert to alanine and alpha ketoglutarate. This alanine will come into the blood stream in the form of blood alanine, and this alanine again is transferred into the liver, and after reaching into the liver, it will again degrade into the pyruvate and glutamate, and this glutamate again lose their ammonia, and this ammonia will enter into the urea cycle, and this pyruvate again enter into the gluconeogenesis and convert it to the glucose, and this glucose again transported to the muscle cell. Now finally, we have already discussed about that from all the tissues we have collected the ammonia in the form of glutamate, whatever it is coming from in the form of glutamine, or it is coming in the form of glutamate, or finally in the liver we got the complete concentration of the glutamate. And in liver, after reaching into the liver, this glutamate will enter into the mitochondria, and in case of when it will reach into the mitochondria, it will again lose its amino group and convert into the alpha ketoglutarate. Whether it will decide whether this ammonia which is generally released into the mitochondria will participate into the biosynthetic process or will participate into the urea cycle, or either if it is not taking part into the biosynthetic process, then it should be excreted out because ammonia is the toxic for the body. So it depends upon the type of organism, whatever ammonia is excreted out in the same form or in other form. Like if we talk about the fishes which is generally lives in water, then in case of fishes this ammonia is taking place in the form of same ammonia. When in case of mammals, this ammonia is excreted out in the form of urea. So before excreted out in the form of urea, this ammonia will enter into the urea cycle. Before entering into the urea cycle, first of all, the ammonia will combine with the CO2 which generally synthesized in the citric acid cycle, and from the citric acid cycle this CO2 combined with the ammonia which is generally coming from the glutamate combined together and form the carbamyl phosphate in the presence of enzyme carbamyl phosphate synthetase, and make the final product carbamyl phosphate. This carbamyl phosphate combined with the ornithine and converted into the citrulline with the help of enzyme ornithine transcarbamylase. Urea cycle taking place generally in both in cytoplasm and in mitochondria. Few reactions take place in cytoplasm and few reactions take place in mitochondria. The synthesis of the carbamyl phosphate is generally taking place in the mitochondria. After the synthesis of the carbamyl phosphate, when it will combine to the ornithine, then it will convert this ornithine to the citrulline with the help of enzyme ornithine transcarbamylase, and this citrulline now is transported to the cytoplasm, from where it is converted into arginosuccinate with the help of arginosuccinate synthetase. Now this arginosuccinate is converted into arginine and the fumaric acid with the help of arginosuccinase, and this arginine is converted into ornithine with the help of the enzyme arginase, and in this step the urea is produced, and this urea is excreted out from the body through the kidney in the form of urine. If you will see, there is a correlation between citric acid cycle and the urea cycle, because in case of the urea cycle we can say that there is the production of the fumaric acid, which is a intermediate metabolite of a citric acid cycle. So with the help of this fumaric acid, or this fumaric acid can enter into the citric acid cycle, and from the citric acid cycle this fumarate will enter into the citric acid cycle, and the aspartate, which generally also an intermediate of the citric acid cycle, is also take part into the urea cycle at the step second. So we can say that with the help of this aspartate and arginosuccinate, these two cycles are interconnected. So we can say that this subtle shunt which is connecting the urea cycle to the citric acid cycle is known as aspartate arginosuccinate shunt of citric acid cycle. Now this is the regulation. If the any metabolic pathway is happening in our body, plant body or any other body, then it should be regulated with the help of so many factors like enzymes, like activators or inhibitors, and some hormones also. So this urea cycle is also regulated with the help of an enzyme, and this enzyme is generally at the step of the first, which is known as the carbamyl phosphate synthetase. At this level we can regulate the urea cycle, and its regulator is the N-acetyl glutamate, which is generally synthesized with the help of acetyl CoA and glutamate, and this N-acetyl glutamate synthase will convert these two substances into the N-acetyl glutamate, which is the activator of this carbamyl phosphate synthetase first. Thank you.